Biology Grade 9, Unit 1, Introduction to Biology. 1.3 The Scientific Method. What is a scientific method? Scientific method involves a series of steps that come up with new theories to explain new findings. The general steps of scientific methods are 1. Careful observation. 2. Ask questions. 3. Literature review. 4. Formulating hypotheses. 5. Experiment. 6. Result and conclusion. Sometimes the result and the conclusion can be a separate step, result first, then followed by conclusion. 7. Communicating findings. What is the hypothesis as steps of scientific method? Hypothesis is a proposed scientific explanations for the questions. Scientific hypotheses should be testable. Testing the hypothesis means doing scientific experimentation. Hypotheses can be tested through scientific experimentation. Any scientific experiment must be duplicated or replicated. What happens to original hypothesis after scientific experiment is very important issue. Original hypothesis can be supported by scientific experiment, or original hypothesis can be rejected by scientific experiment. Result in making conclusions about the findings. The hypothesis can remain as possible explanations for how things work if it is correct in light of the new information gathered during the experiment. However, if hypothesis is not correct in light of the new information generated during the experiment, scientists reject the original hypothesis and try to come up with alternative explanations, which means new hypotheses. Communicating the findings. This is the last step of the scientific method. Scientists can communicate their findings through scientific journals, posting in websites, and using communication media. Now, let us take typical examples and try to describe what the scientific method is. Suppose you always see that bean seeds germinate faster in hotter places compared to colder places. So let us work out scientific methods. Careful observation is the given statement. Ask question step. Why do bean seeds germinate more quickly in hotter places than in colder ones? Literature review. So we have to read anything about seed germination. Next is formulate hypothesis. Take this example. In hotter places, a higher temperature speeds up seed germination. Yes, this is a testable hypothesis. Let us look at this example. In colder place, something inhibits germination of seeds. This is not a hypothesis. Something cannot be tested by scientific experiment. So it could not be a hypothesis. Do experiment. Place a paper towel in 6 clean glass jars. Place 5 bean seeds in each jar. Keep 3 jars in kitchen and 3 jars in sitting room. Here, each jars have 5 bean, and jars replicated 3 times. Provide water and sunlight. Measure the shoot length of the seedlings every day of germination. First day of germination, second day of germination, third day of germination. This is the recorded data. Germination days, first day, second day, and the likes. Seed germinated in chicken in 3 jars, jar 1, jar 2, jar 3. Chicken germinated in sitting room, also germinated in 3 jars, jar 1, jar 2, jar 3. Remember, in each jar, we placed 5 seeds. Here, the X shows the seeds failed to germinate. For example, for seed germinated in chicken, in jar 1, 3 seeds germinated and 2 seeds failed to germinate. In jar 2, 2 seeds failed to germinate and 3 seeds germinated. Jar 3, 2 seeds failed and 3 seeds germinated, and the likes. Results and the conclusion. Now, the data for shoot length should be average and become result. So the result is represented in 2 form, in table form and in graph form. In table form and in graph form. Now, first day, bean seed germinated in chicken have 3 centimeter shoot length. On second day germination, 4 centimeter length. Third day germination, 6.2 centimeter length. Fourth day, 9.4 centimeter length, and fifth day, 11.5 centimeter length. For bean seed germinated in sitting room, the smaller values were observed. The graph also shows the same value. Blue shows the shoot length of seed germinated in kitchen, and brown shows the seed germinated in sitting room. As you see, the blue bars are longer than the brown bars in each case of bean seed germination. So conclusion, bean seed germinated in chicken had longer shoot lengths than bean seed germinated in sitting room throughout germination day. considered. In kitchen, a higher temperature speeds up seed germination. This was the hypothesis, and this hypothesis is supported by the experimental results. The last step of scientific method is communicating the findings by publishing in journal or posting in websites or using communication media. Thank you.